Dr. Sathyan.
Thank you, Kelly. Let me share my screen. Hi. Thank you, Kelly, for the introduction. I'll talk about our paper, Interpretable AI for Biomedical Applications. This was co-authored by Abraham Weinberg, who is on the attendee list, and Dr. Cohen. This paper talks about using explainable AI methodologies, that is LIME and SHAB, on the breast cancer Wisconsin dataset. This is a very popular dataset on the UCI data repository. So the objective here is we have a breast mass, and we are extracting features from the breast mass, the digitized image of a breast mass. And for each of the cell nuclei present in that breast mass, we extract a certain number of features. In this data, we are considering 30 features that are listed here. I'll go over them in a minute. But the objective is, based on those 30 features, first of all, can we accurately classify them as benign or malignant for each of the 569 data points that we have in the dataset. So the input features, that is, the 30 features include the radius, the texture, the perimeter, area, smoothness, compactness, concavity, concave points, symmetry, and fractal dimensions. And we are extracting 3 statistical factors for each of the 10 variables, and that includes the mean, the standard deviation, and the worst value, which is the maximum value for each of the 10 features. So those are the 30 input features that we are considering for the neural network model. And it will have 2 classes, benign and malignant. So this is essentially a classification problem. And this is I'm considering a deep neural network here with 3 layers. The first one has 72 neurons, second one has 36, and the last one has 18. And then we have the output layer with the 2 classes, benign and malignant. So this is a standard classification problem. We consider the binary cross entropy loss during training. And the data set with the 569 data points, we use random 80-20 split. And this is the test accuracy obtained over the 20% validation set. So that's obtained at 96%, which is very good. But that is not the focus of this paper. It is more to understand the use of explainable AI techniques. I'll start with the LIME method to see how we can explain the decisions made by the trained neural network model. So let's go over LIME first. So LIME stands for Local Interpretable Model Agnostic Explanations. So how this works is for each data point that we want to explain the outcome for. So we have the data point, and then we have some outcome that's obtained through the neural net. So it will say whether it's benign or malignant. So now we want to know why it's benign or why it's malignant. So for each data point, we perturb the observation n times. So essentially, we obtain n number of neighboring points around this data point. And if we consider that as a tabular data, by evaluating each of these n data points using the trained neural network, we will get, again, benign and malignant classes. So if we consider that tabular data, then we can create an explainer to explain why this model is making this prediction. So we have the original model making the predictions on the n neighboring points that were picked out of the neighborhood of the original data point. And then we calculate a distance from the perturbed points to the original observation and convert that into a similarity score. Then using this data set, that is the n new data points, we can use a simpler model, that is a linear model, on the perturbed data and explain the outcome. Because with a linear model, you have the coefficients, the A1, H1 plus A2, H2. It's very easy to explain that process. So using that, the coefficients, we can evaluate how important each feature in the data set was for that particular data point. So based on the feature weights, we can provide the explanations. So I'll show you three cases from the test data. So here is one of the data points. So this is the feature vector with the 30 values. And you can see this plot. Essentially, this is the outcome from the line analysis. And in this case, this is classified as malignant. This is correctly classified as malignant by the neural network. And what this is showing essentially is that input number 21, 27, and 24 are the most dominating features in this for this particular data point. And that is the contribution factor, 0.28, 0.22, 0.18. And what you can see here is essentially higher values point towards malignancy. And then there are also some variables that kind of push it towards benign, but still these kind of overpower, the top ones overpower these ones. So the eventual outcome from the neural network is correctly predicted as malignant. And this is the Lyme analysis shows why or which features are causing the neural network to classify it as malignant and why it is so. So here is another case where the neural network is predicting the outcome as benign. And here you can see all of these features. So input number 20, SIDS, 22, 7, 26, 21, these are all contributing towards this being benign. This is truncated, but in this, you can see that this is the first variable number 28, that kind of point towards malignancy. But even then, the contributing factor is quite low at 0.07. Moving on to the last data point that I'm considering here. Again, this is a case where the neural network classifies this as malignant, and you can see the contribution factors for these variables. So these are the dominant variables, 20, 21, 22, 6, 27. Those are the dominant variables for this prediction. And you can see that these are pretty high compared to the previous ones too. And in this case, there is no variable that is pushing it towards benign. So this is very clearly a malignant case. So the other method that I considered is the SHAP. So SHAP is short for Shapley values. It comes from a concept from game theory. So in game theory, the objective is you have a set of players that need to cooperate, and that would decide the outcome of the game. So in this case, the game is basically reproducing the outcome of the model. So the prediction from the model is essentially the game, and the players are the different features. So how the different features interact with each other to produce that certain outcome. So Shapley values basically provide a way of representing the contribution of each player, which in this case is the feature, to the game, which is the output prediction. And once we have the effect of the combination of features, then we can understand, again, why certain predictions are made. Now, the SHAP process is a little more detailed. So there are faster SHAP processes now, but I'll talk about the standard one here. So if we have F number of features, then we consider 2 to the power F coalitions, because we are essentially considering each combination of features. So you can see here at F equals 0 layer, we don't have any feature. So that would essentially point to the mean of the outcome variable, whatever that is. So this is the null model, which doesn't consider any feature, and it just outputs whatever the mean of all the cases in the training set is. And at F equals 1 level, we are considering each individual feature. So in this example, it's age, gender, and job, and they're considered separately. So each of these are separate models. One considers just the age and makes a prediction, makes an outcome prediction. Then another model just takes in gender as the input and makes a prediction. And the last one takes in job as the input and makes a prediction. Then here is the second layer, F equals 2. Here it combines 2 inputs at a time. So age and gender, and then you have the outcome. Age and job, you have the outcome, and gender and job. And since this is, in this example, we are only considering 3 variables. F equals 3 is the last layer, and that would be age, gender, and job. So we have 2 to the power f, that is 2 to the power 3 would be 8. So 8 separate models that need to be trained. And then based on the marginal contribution of each of the models, we can evaluate the contribution of each feature towards the overall outcome. And that is referred to as the Shapley value. So if the Shapley value is high, then that means that particular feature is contributing a lot to the outcome. Now, in the case of Shapley, it provides these summary plots over the entire data set. So this provides, so instead of trying to see data point by data point, we have a good summary for the entire data set. So you can see here some of the ones that are contributing the most. So most of them relate to the worst values or the maximum values of the radius, the concave points, the texture. Those are the important ones that are contributing to the different predictions. And at the bottom, we have the standard deviations of fractal dimension, the worst area. Those are the ones where you don't see too much variance here, and it's contributing less to the model. The x-axis here is the SHAP value for the benign case. So as you go towards the right, as the SHAP value increases, that means the model is making a benign prediction. So you can see here the radius worst, and these are color coded. So you can see the red values, they point to higher values of these variables, and the blue ones point towards lower values. So you can see a clear distinction in most of the variables. As you have lower values for the variables, the outcome happens to be benign, and as you go to higher values for those variables, the outcome is malignant. So it provides a better summary over the entire data set. And there are additional plots that come with the SHAP package in Python. So here is another one that shows the dependency between two features. So you have to define which two features you want to analyze. So in this case, I'm analyzing the texture mean and the texture worst. And I'm trying to correlate it with the SHAP value for texture mean. So what this shows is that as the texture mean increases, the SHAP value is decreasing. So that means this is for the malignant case. So as the texture is the mean of the texture is increasing, the SHAP value is decreasing, which means that the model is more likely to make a malignant prediction because the SHAP value here corresponds to the malignancy. And you can also see that these are color coded. So that shows the correlation between texture mean and texture worst. And in this case, as texture mean is low, texture worst is also low. And as texture mean is high, texture worst is also high. So that's kind of, there's a linear correlation there. And that can be seen from the dependency plot. And in this case, while the SHAP value and the concave points mean they have a similar relationship, the relationship between the mean of the concave points and the symmetry standard deviation is not immediately apparent. You can see a mixture of colors here. So that's the advantage with dependency plots. You can also get kind of a correlation between how different pairs of features are interacting with each other to produce that outcome. So just to summarize the presentation, basically I have considered two explainable AI techniques, LIME and SHAP, and I've analyzed that on the breast cancer Wisconsin dataset, which is a very popular dataset on the UCI repository. But these can be used on any neural network. It doesn't have to be neural net. It can be any model. These are model agnostic techniques. Now, in this case, we only have 30 features, but as you have more number of features, this becomes more complex, but it still provides a way to understand how or explain why a model is making certain predictions. So that's it from my side.
So if there are any questions, we'd like to answer them. We have time for one question from the audience, from the participants. or from the panelists? Anyone? You want to challenge Anoop? I'd invite the co-author, Avi, if you want to make a statement about this work or emphasize, underscore a certain point. Yeah. Could we allow
I have a
Avi to
question.
speak?
Hello, Kelly, do you hear me?
Yes, we do, very clearly. Please go ahead,
Okay.
Avi.
So first of all, I would like to thank you for presentation. It was very clear. I'm not objective in this case, but I think that the presentation was very, very clear. And I would like to ask maybe Anoop, can you maybe emphasize the differences between the approaches of SHAP and LIME in the contribution to explainability of the models and neural networks?
Right. So based on my understanding, so LIME is mostly like you're trying to explain individual predictions on individual data points. And it uses the neighborhood of that data point to make that explanation. Whereas SHAP tries to do this on a global level. It considers the entire data set, and essentially this summary plot shows clearly the explanation for why the model is making those certain predictions over the entirety of the data set. And it's making those explanations based on how different features pair with one another. And we are considering different combinations of those features, as you can see from this plot. So that's, I think LIME is more local and SHAP is more of a global explainer.
Thank you very much. I think that the future of explainability and interpretability of neural networks and AI is the basis relies on the SHAP and LIME, and the future has many combination between them, but this work really exemplify and demonstrate the importance of these tools in a use case that is very, very important to the field.
Thank you very much, Avi. And with that, I would go on to my next speaker.